Assalamualaikum class 11 students, higher secondary school, Langate, and we will continue the topic bacterial structure. And in bacterial structure, I have taught you several structural features, and today I will discuss some of the important, the significant features related to the bacterial determinants. Another important determinant of bacterial ribosomes. The ribosome, if this is bacterial cell, it contains several. It contains enthalments. It contains these spherical bodies, sometimes irregular bodies, which are sites for protein synthesis. Sites for protein synthesis, these are called ribosomes mean fragments. Ribosomes mean fragments which are mostly formed of protein and some carbohydrate components. So ribosomes are spherical bodies present in the protoplasm of the cell, which are the sites for protein synthesis, and each bacterial cell is thought to contain many ribosomes. Containing many ribosomes, and E. coli is thought to contain 10,000 ribosome molecules. Ribosomes. Also, the ribosomes in bacteria in bacteria are 100 angstroms in diameter, and their sedimentation coefficient, when it is process of centrifugation or the separation of the molecules from a compound. And this it is the Selberg's unit, which when he proposed for the sedimentation of ribosome in case of the eukaryotic and prokaryotic cell. You will go through the books or in higher class to go and read yourself what are the units and subunits of ribosomes. This question has many times been put in exam, and I was facing my entrance exam in MSc level and also in PhD level. I found many of the questions of Selberg's unit. So, sedimentation coefficient of the ribosome of bacterial ribosome 70. S is the unit given by Selberg. So S, it is subunits are 40S and 50S and 30S and 30S. So this is all about the ribosome found in the bacterial cell. Next important feature of the bacterial cell is nucleoid. Nucleoid is next. Nucleoid. Important bacterial component. It is important bacterial component, or it is also, we can say, the genome of bacteria, or the genetic material of the cell. Bacteria or genetic material of the cell. In case of this bacteria, the genetic material is DNA. DNA is genetic material, and it is naked. Naked means in eukaryotic cells, the genetic material remains enclosed within a membrane that is called the nuclear membrane. But in case of these bacteria, the nuclear membrane is absent and the genetic material is naked. And when the genetic material is naked, it is called nucleoid. Also, one of the important features of the genetic material, this naked genetic material, is that these histones, in case of higher eukaryotic cells, the histones, DNA molecules are present or associated with histones. Histones are special kind of protein. Histones, non-histones are special kind of proteins which DNA molecules are present. But in case of prokaryotic cells, that's bacteria, DNA molecules are not associated. DNA molecules are not associated with histone molecules, and as such, chromosome formation is absent. Chromosome formation is absent. That chromosome in the case of bacteria are not found. Also, I can say the genetic material or the DNA in the bacterial cell. Bacterial cell, that is genome of bacteria, is a single circular double-stranded DNA. Circular double-stranded DNA occupying much of the portion of the cell, and as such it is structured in the cell, it is adjusted in the cell, it is packaged in the cell in a structured way so that it can accommodate in the cell. If it is in loose form, then it is impossible for the cell to accommodate whole DNA. That's about the packaging which you need in higher classes. So circular bacterial DNA or bacterial DNA are circular molecules of double-stranded DNA. Only one double-stranded DNA is present per cell. This is called the DNA or the macrogenome. DNA of bacteria, macrogenome. DNA replicates once the cell has to undergo the process of this reproduction and asexual reproduction, that is mitosis, and this is the circular DNA present. This DNA has many. I'll tell you, the gene locus means a portion of the DNA which where the specific kinds of genes are present, and each DNA has many loci, and each loci contains many genes. DNA has many. DNA in bacteria has many gene loci, locus, plural of locus is loci, and each loci contains many genes, and it is estimated that the gene locus in case of the E. coli has 4,000 genes. Gene loci of E. coli contain 4,000 genes. Thank you. This is sufficient for today. And if my cameraman is my student, if she met me after namaz, I will try to deliver you Zoom class at 3 p.m. Thank you very much. Till further.